Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show, bringing another really awesome guest today involved in science and technology that's creating a better tomorrow for all of us. Today on the show, we have the honor of being joined by Dr. Kilian Kelly. He is the Chief Executive Officer and Managing Director of Cynata Therapeutics. They are a stem cell and regenerative medicine company known for their proprietary Cymerus platform that's focused on a scalable and consistent production of mesenchymal stem cell-based therapies. And unlike traditional processes, which rely on a lot of donors, their manufacturing process ultimately assures that these cells can be scaled up in virtually limitless quantities from a single donor. The company has completed a phase 1 study in graft-versus-host disease and diabetic foot ulcers. They also have a number of phase 2 and phase 3 clinical trials in progress. Dr. Kelly has over 2 decades of experience in biopharma R&D, 15 years focused on MSCs, and while at the company has overseen all stages of their platform, including the Cymerus induced pluripotent stem cell derived technology, and ultimately the company is actually the first that has completed a clinical trial of any iPSC-derived product worldwide, interestingly. Dr. Kelly previously has held positions at Biota Pharma, Mesoblast, Kendall, as well as Amgen and AstraZeneca, has his master's pharmacy degree, Robert Gordon University in Aberdeen, went on to do a Ph.D. At Strathclyde University in Glasgow, and is a graduate of the Australian Institute of Company Directors in Melbourne. He's also a member of the International Society for Cell and Gene Therapy, where he serves on their Asia Pacific Industry Committee, the International Society of Stem Cell Research, and he's part of their best practices working group for pluripotent derived therapies, as well as the Royal Pharmaceutical Society. A lot to get into today, a lot of really cool topics. Honored to have him with us. Dr. Kilian Kelly, thank you so much for taking time on the show today.
Thank you, and really looking forward to the conversation.
Yeah. I love all these topics. I'm really looking to get into all this. Before we start though, and get into anything stem cell, I'd love to hand things over to you just to further talk a little bit more about your background, because one of the really interesting things I saw, your Ph.D, you're in essence a pharmacokineticist, and I was mentored by one years ago early in my career. I have amazing respect for anyone that gets into pharmacokinetics at any level, involved in studying drug absorption and interesting forms of paracetamol and so forth. Talk a little bit about the early days, why you got involved in the industry, and a little bit of sort of the pre-cell therapy stuff when you were working on what we think of as traditional drugs nowadays, before we get into biologics.
Sure. Yeah, well, you're absolutely right. So I certainly didn't start out as a cell therapy guy. I was, originally qualified as a pharmacist, spent a year working in a hospital pharmacy, and then went and did a Ph.D, which is very much a typical, pharmaceutical sciences Ph.D. The University of Strathclyde, and as you said, studying ways to try and alter drug absorption profiles, and my PhD really involves specifically trying to work with oral formulations to somehow alter their gastrointestinal transit. So, as you would know, the length of time a formulation takes to go through the gastrointestinal tract has a major impact on the drug absorption profile, the extent, the rate, and so on. So we were trying to find ways to actually add excipients, mostly to oral formulations, to either accelerate gastric emptying or the reverse, in fact, in some cases, to delay it. So it was really interesting. As I suppose you've alluded to, has very little to do with what we're doing right now at Cynata. But what it did was it gave me some really, like any PhD, I suppose, it gave me a lot of experience that ends up being useful in perhaps unexpected ways. But one of the things I did as part of this PhD was I did get involved in actual human studies because it's very difficult to study gastrointestinal transit in a human without actually studying it in a human, right? So there's no real very good models for that. So what we did was we had some like healthy volunteer studies where we were like radio labeling these formulations and studying how they dissolved and left the stomach and all of that sort of stuff. But I guess where that sort of became relevant to my later career is it kind of gave me insight into how you actually do a clinical trial, right? Just the logistics of it, the fact that you've got to get the various ethics and regulatory approvals, you've got to write protocols, you've got to be really specific about how you're doing things. There's the need to obviously to get people's consent and all of that sort of stuff, and then to actually find the people who are willing to participate in the trials. And that really, I suppose, was my grounding in the world of clinical trials more generally, which is sort of where my career went after I finished my PhD. So, while my PhD was really interesting and I enjoyed a lot of it, by the time I'd finished it or come towards the end of it, I'd sort of reached a point where I thought I don't really want to be an academic for the rest of my career, so I'm going to go and move to the dark side into industry, and so that is exactly what I did, and where I was really then interested was on the sort of drug development side of things. So I initially my first job after my PhD was with AstraZeneca in England. Initially in a regulatory affairs role, but regulatory affairs really is I suppose a supporting function behind drug development projects. So that it is involved in the clinical trial processes. Also, ultimately, of course, any drug development program, the aim is to get regulatory approval at the end of the day, right? That's what we're doing. So regulatory affairs gave me a really sort of interesting, overview of that whole process. And I spent a few years with AstraZeneca, and then moved to Amgen, also in the London area. And at that point, I was working on, being Amgen, of course, it was monoclonal antibodies. I worked on a drug called denosumab, which at the time was in phase 3 trials for osteoporosis and also various other indications, including bone metastases and so on. Now, of course, that product is on the market and has been for some time. But that, I guess, was a little bit of an evolution away from, your small molecule drugs, which is what I was doing in AstraZeneca, into the world of biologics. But I still had nothing to do with cell therapy at that point. And really the reason or sort of the path I took that led me to where I am today. So as you may have picked up, I originally grew up in Ireland. Did my university education in Scotland. Then I was in England. But while I was in Scotland, I met my Australian wife, who eventually persuaded me to move to Australia. So in 2008, we moved to Melbourne, and that was certainly not something that I have any complaints about. It's a fantastic place, and I've been in Melbourne for the last 17 plus years at this point, and we now live here with our teenage children. But one consequence of the move here was I sort of had to pivot career-wise a little bit, because the big pharma guys, they have a presence here in Australia, but it's not really they're not leading R&D from Australia as a general rule, right? So the AstraZeneca and Amgens in the world, they'll have, obviously they'll have sales and marketing people here, and distribution, and maybe some manufacturing and so on, but they don't really have the sort of roles that I was, at that point settling into. So I had to find something a little bit different. And the first role I got here was with a consulting group at Kendall International. a regulatory development and commercialization consulting group, and most of their clients were small biotech companies, because that is really the what most R&D here in Australia is all about. It's small biotech companies. And that's really what gave me a step into that small biotech scene. And then after a couple of years of working in that consulting role, I was recruited by another stem cell company also based here in Melbourne called Mesoblast. And I'm sure you'd be aware of them because they've quite recently got an FDA approval, which is fantastic. But they also are developing mesenchymal stem cell, or MSC-based therapies. So that was really my when I kind of moved into that space. That was back in 2009, so quite a while ago now. And apart from a brief stint at Biota between Mesoblast and Cynata, I've spent really all my time since working on MSC-based therapies. So I guess it's a career path that was, I'd be lying if I said back in the sort of late 90s, I decided this is exactly what I'm going to do. That's really not what happened, but I think that's probably rarely true for anybody. I think you kind of you evolve as you go through things, and you take opportunities as they come up, and often that works out well. And I think in my case, it certainly has.
Absolutely, absolutely, and I appreciate you taking us going through that path, because I very much enjoy hearing about that evolution of our guests. So very cool. Before we get into everything that's going on at the company, and we talk about Mesenchymoangioblasts and sort of the IPS technology that you've worked with. Take us again. We've talked a little bit about MSCs on the show over the last couple of years, but let's refocus on them. Just in terms of what they are and what they currently do, sort of because they're sort of the workhorse of the stem cells. Obviously, we heard a tremendous amount a couple decades ago about embryonic stem cells. We hear a decent amount about iPS cells nowadays, but it's really the MSCs that, if you pointed out, I mean, we've gotten approvals based on them. They are not, totipotent, they're the multipotent, so they're capable of becoming lots of things that we're interested in, from bone to cartilage to muscle. And we've had a decent success story in terms of safety and efficacy to date. Walk us through a little of the history and a little, you mentioned, you were a Mesoblast, you experienced the clinical development of them. Key learnings, takeaways in terms of, what it has been like in taking this concept of, this multipotent cell a couple decades ago to an actual therapeutic product.
Yeah, so you're right. Like MSCs, I think, have by a long way been the most widely used cell therapy modality, right? And there's obviously lots of different types of cell therapies being used for all sorts of purposes. But in terms of the number of patients who've received cell therapy, MSCs are by far and away out. And the history is, so, the first clinical use of MSCs was in, I believe, 2004. So leading up to that, obviously, you have, all sorts of basic research going on and so forth. And it was known that MSCs, of course, they are naturally occurring cells in everybody's body. It's known that they have a number of potentially very useful properties. One being, and this was probably more of a focus in the early years, but the fact that they can differentiate into other cell types, as you mentioned. So, in particular, they can make bone, fat, cartilage, and so on. But what they can also do, and I think the understanding has evolved in the over that period is, and this is really where their use is mostly, is that they have immunomodulatory and anti-inflammatory properties, and they also promote tissue repair and regeneration. So it's not necessarily a case of the MSC itself becoming, for example, a cartilage cell and so on. It's about promoting the repair of other tissues that they come in contact with. And so the clinical history then is really interesting, because the first documented use of MSCs in a human patient was in Sweden, and as I said, I think it was 2004. Led by Katarina LeBlanc at the Karolinska Institute. And the situation was, they had a young child, I think it was a 9-year-old boy, who had graft-versus-host disease, had failed all existing treatment options. He was really at an end stage, not expected to survive. And the team there had this idea that MSCs could potentially be useful in these sorts of patients. And as a sort of a compassionate use, last chance option, they got approval to administer MSCs to this boy. In this case, the MSCs came from bone marrow donated by the boy's mother. And what they found was extraordinary because the boy made a complete recovery, right? So, what an incredible good news story for a start. But it was more than that because it really showed that there is actually something in this. And so that they then published this case report in Lancet at the time. And that really led to this explosion of MSC-based research and lots of trials being started for graft-versus-host disease initially, but then in many other conditions, because of course, properties like immunomodulatory effects and anti-inflammatory effects have very broad applicability, right? So many disease processes involve some sort of immune dysfunction and or inflammation, right? So if you can control those things, then you have a treatment that has many potential uses. And of course, promoting tissue repair and regeneration is also, very broadly useful. So from then, things really kicked on, and initially it was mostly academic trials in a lot of them in Europe, but then we started to see some companies getting in on the act as well. So there was Cyrus Therapeutics in the US who started running trials with a commercially produced MSC process, again derived from bone marrow donors. The difference with what they were doing was, of course, being a commercial operation, they were trying to produce much larger batches of product with a view to sort of off-the-shelf usage. In contrast, the academics were mostly either using sort of autologous, patient's own cells, or maybe related donors or even unrelated donors, but making, really just treating maybe a handful of people from one donation, right? So we had a lot of activity going on there. And then after Osiris, many other companies started to pursue MSC-like therapies as well, one being Mesoblast, which I mentioned earlier, Athersys also in the US, and lots of others, Pluristan, numerous companies around the world, really. And the upshot is, I think today, and the last time we looked, in total, there's been more than 1,700 clinical trials using MSCs registered on clinicaltrials.gov. So that's quite extraordinary. And the number of different conditions is also massive. I mean, I think I've heard a figure of more than 100 different clinical indications that MSCs have been used for in clinical trials, which is, again, quite extraordinary. But what sort of has become clear over that time, though, is the idea of kind of taking what those academics did and getting bone marrow donations and using the cells from that, and then turning that into a huge commercial process. That's not as straightforward as it may have seemed initially, because MSCs do have the ability, of course, just to explain for background, if it's not obvious, the way that they do that is they take a bone marrow donation, for example, they isolate the MSCs from that donated bone marrow. But of course, bone marrow consists of all sorts of different types of cells, and most of them are not MSCs, right? So there's only a small proportion of the cells that are MSCs. So when you complete that isolation process, which is, fairly straightforward, and there's various ways to do that, you can get MSCs, but not very many. So perhaps from a bone marrow donation, you might get about 20,000 MSCs. And to provide a bit of context there, you need usually hundreds of millions of MSCs to treat one patient, right? So if you're trying to make a big batch where you can treat thousands of patients, you need billions or perhaps even trillions of cells. So the way they do that is through culture expansion, just growing cells. MSCs will grow quite well in culture. They will reproduce, and you can turn 20,000 cells into quite a large number of cells. But what started to become clear over time, and there are now many, many publications on this, and I stress not from Cynata, who people might think of a vested interest in saying this, but from all sorts of groups in academia around the world, it's very well established that when you extensively expand MSCs, they start to change, right? So initially there was a view that, eventually they will reach senescence and stop growing anymore, and that's where you have to stop. But then what sort of became clear over time is it's actually much more of a continuously changing process. And what you can see then is even long before they reach senescence, they start to lose functionality, basically. So a lot of the properties that you're trying to harness start to, well, dissipate or even go away altogether. So the problem then that became clear is you kind of have to be careful how much you grow these cells. And what that means is then really you have a limit in how many cells you can make from one donation. So you have to keep going back and getting new donors, right? Which again, that sounds okay. We can do that. However, there's a couple of problems with that as well. One being just a logistical problem. If you were trying to make these cells at huge scale, right? How many new donors do you need, and can you do that? Especially when we're talking about donating bone marrow, for example, or even adipose tissue. You know, that it's a bit of a step beyond donating blood, right? It's a more invasive procedure, harder to persuade people to do that. But perhaps the even bigger problem is, again, this has become clear over the last sort of 20 years, is that there's huge variability between donors. So if you're relying on new donors all the time, it's going to be very hard to have a consistent product. So that's sort of the challenge that sort of became clear. And although there's been lots of progress made in donor-derived MSCs over that time and lots of clinical trials, not all of those clinical trials have been successful. And, I believe, and I think a lot of people believe, that one of the reasons for those sort of, if you like, mixed results is the challenges with making the cells consistent. Because if you don't have a consistent product, you're not going to get consistent results.
Yep, absolutely. And yeah, thank you for laying that history there. So amazing potential of MSCs. We got a production issue. We got a scalability issue. What else is happening at the same time? Well, and then again, we'll take a little trip back. 1998, Jamie Thompson, University of Wisconsin, discovers the embryonic stem cell. Decade later, he's working on iPS, and we talked about Shinya Yamanaka and that whole history, but nonetheless, induced pluripotent stem cell work also happening there at the University of Wisconsin-Madison. Your founder, Professor Sluckman, comes up with a concept that we can take this iPS concept, which I'm plucking off my skin cell, I'm turning it back in time with my magic, and I can make anything from it in large quantities. And because it's a very early stage stem cell, we can do that over and over without, as you were saying, running into a lot of those problems with senescence and so forth. And we want to create an intermediary cell known as the Mesenchymoangioblasts, which between the iPS and the trillions of cells we want is sort of an intermediary cell that does a lot of interesting things for us in that process. Take us into that part of the history, because this is very important, obviously, for the company, for the Cymerus technology, and what ultimately that platform is all about.
Yeah, absolutely. So you're right. This all goes back to the University of Wisconsin-Madison, and Jamie Thompson and his group, and Igor Slukvin, who, as you said, is our founder, was part of that group. So he was heavily involved in all of that work that was done there around embryonic stem cells, and then subsequently iPSCs, which, as you say, are a sort of a man-made version of embryonic stem cells, where they take adult cells and reprogram them. And one of the, there's obviously lots of different reasons why people were interested in that whole pluripotent stem cell advance. But one of the things that's key, especially for us, is that pluripotent stem cells, they really can be reproduced in culture to an effectively limitless extent, right? So I mentioned a moment ago that that's not really true of MSCs, because they start to change and then they reach a point where they just stop reproducing together. Pluripotent stem cells, that doesn't certainly happen within the sort of level of expansion that would matter to a company like us. And to give you an example, a group published a paper a few years ago where they showed that they could expand iPSCs by a factor of 10 to the power of 72, and they were unable to detect. Yeah, right, exactly. And they were unable to detect any sort of perceptible changes in the iPSCs despite that enormous amount of expansion. So what that tells us is, in practical terms, we can make as many, once we have an iPSC line, we can make as many of those cells as we want, really. be in real terms. Now, people sometimes say, oh, well, there must be a limit somewhere. And yeah, sure, there probably is, but it doesn't really matter because it's so far beyond where we need to get to. So that's for us. The really important point is that if you have a iPSCs, you can make an effectively limitless number of cells from one donor, right? So now, if you can do that, then the question is, can you make the therapeutic cell that you are interested in from iPSC? Because if you can do that, you now have a way of making an effectively limitless number of your therapeutic cells from one donor. And that's exactly where Igor's discoveries come into play for us. So he, as you pointed out, he identified this intermediate cell type called the Mesenchymoangioblasts, which is basically a common precursor to the mesenchymal and endothelial lineages. So it's an earlier stage on the sort of development pathway towards MSCs. And other cell types that should be mentioned, but from Cynata's perspective, it's the MSCs that we're focused on. So what he found was that he identified this process then where we could have a directed differentiation process. In other words, we can take iPSCs and we can basically cause them to differentiate into mesenchymoangioblasts, and then from mesenchymoangioblasts into MSCs. And what that means then is we have, when you put that together with the iPS cell technology, what we have is a way of taking one donation from one donor, making an iPS cell bank, and then just basically churning out MSCs from that iPS cell bank to an effectively limitless extent. And those MSCs then are, first of all, they're all coming from the same starting material from the same donor. So we have that consistency at the start of the process. Another advantage is that we don't need to excessively expand the MSCs. So once we turn the iPSCs into MSCs, we don't need to excessively expand them. And the reason for that is, I mentioned earlier on that if you start out with bone marrow, you might only get 20,000 MSCs from your bone marrow donation, right? We don't have that problem because we can make as many iPSCs as we want. So we get the scale at the iPSC stage, and then we turn those iPSCs into MSCs. So it's a way of generating vast numbers of MSCs without needing to excessively culture expand the MSCs themselves. And what that means is we also avoid those problems I mentioned where the MSCs start to change through that expansion process. So what we end up with is a sort of a minimally expanded MSC product, which is probably much more similar to those academic bone marrow derived MSCs, you know, where they're just basically treating one or two patients from each donation. Our MSCs are probably more similar to that in an expansion sense. Just for completeness, there are some differences between MSCs from different sources. So I'm not trying to say our cells are identical to bone marrow MSCs because they are not. But from a sort of an expansion perspective, and sometimes people use the term sort of, you know, younger MSCs, which really means that they're, you know, they haven't gone through as many divisions, if you like. So that's sort of what we are producing, and we're able to do it, as I said, then in a very consistent way because we aren't changing our starting material, and secondly, we're not going through this very extensive culture expansion process at the end. So we have then what we believe is a way to make an effectively limitless number of MSC doses that are very consistent batch to batch. And we've generated a lot of data now to really verify that is indeed the case. And, you know, we believe that that's really the way forward. And I should mention that we're not the only ones, as I know you're aware, we're not the only ones who've come to the view that iPSCs have huge potential for these reasons. There's lots of other companies now using iPSCs to make all other cell types, and in many cases they're doing it for the same reasons, because it's a way to really sort of industrialize the production of cells as opposed to sort of harvesting cells from nature, if you like. So that's sort of how I would.
No, it's a very elegant process again, and clearly, you know, along with versatility and manufacturing scale up and all that, you know, you've obviously done a lot of safety work because you've your clinical portfolio is quite impressive, and I'd love to just to take some time to go through a little bit of what you're experiencing here, some of these different indications, because I know you're at all different stages of clinical development. I know a really great place to start, and it's a very hot topic, is of course GvHD, or graft versus host
Mhm.
disease. We talk more of the opposite, I guess, on the show, in terms of host versus graft. We get an organ, and it's rejected.
Mm.
In this case, this is a very severe complication when a stem cell or a bone marrow transplant is infused. It can go wild in the patient's system and start ripping up all parts of the body, various organs. Talk a little bit about you sort of the clinical program that you've put together, and I know you've been executing on this for a couple of years now, but at the same
Mhm.
time, you mentioned these cells, immunomodulation, anti-inflammatory, interesting regeneration capabilities. Talk a little bit about sort of the pharmacology that's going on here, or the putative pharmacology that's happening here, you think, when you're seeing, obviously, significantly reduced GvHD in the clinical work that you're doing.
Yeah, so especially when it comes to GvHD, this really centers around the immunomodulatory properties that
Right.
that the cells have, and I think it's important to stress that we're talking modulation as opposed to suppression.
Right.
So there's lots of drugs, of course, that are immunosuppressants, and some of those can be useful to an extent in conditions like GvHD, but they often cause more problems than they solve because they're a very blunt tool that's just strongly suppresses the immune system, and of course that has consequences. We have immune systems for a reason, and if you stop your immune system functioning properly, then you're obviously at risk of infections and indeed increased risk of cancer and so on. And some of those immunosuppressants, in fact, have their sort of direct toxicities as well. So what's important to say here is that MSCs, they're not immunosuppressants in that sense, but what they do is modulate, or basically help balance the immune system again. So a key way that MSCs work is that they, well, first of all, more broadly, they work mostly by paracrine. So they release all sorts of cytokines and other proteins and so on, which have effects on other cells around them. That's basically how they work at a high level. But one key way that they work in terms of immunomodulation is by secreting something called indoleamine dioxygenase, or IDO. And MSCs secrete that constitutively, like even in a sort of a resting state. Now, interestingly, the specific MSCs that we make express that at a higher level than donor-derived MSCs, we've found, which is useful. But what is perhaps even more relevant here is that when the MSCs are then exposed to an inflammatory environment, so for example, when they come in contact with interferon gamma or TNF alpha, the level of IDO secretion is very significantly upregulated. And the reason that's important is that IDO then helps to suppress the proliferation of CD4 positive and CD8 positive T cells, which is really what drives GvHD in particular, right? So they help to basically suppress that fundamental mechanism of GvHD. They also, on the other side of the coin, they actually help T regulatory cells proliferate, which again helps to bring things back into balance. And one thing I should say here is that what I've just explained obviously is one part of their mechanism. MSCs do have quite a complex and very multifactorial mechanisms of action, because they secrete a whole lot of different proteins and other bioactive molecules. So there's still lots of sort of debate and lots of papers coming out all the time about exactly what they do. But we believe in GvHD in particular, a key part of it is that IDO driven mechanism. And so in terms of how we moved our product into the clinic, I mean, one thing I obviously have to acknowledge is, and as I mentioned earlier, using MSCs to treat GvHD wasn't our idea, right? That was already being done since way back in 2004 in Sweden. And since then, other companies as well have got involved in that, as well as other academic groups. So we had the ability to sort of build on what was already out there. In other words, that there was already a reasonable sort of acceptance that MSCs are useful for treating GvHD. So in terms of our development program, the key things that we had to do was, first of all, show that, well, we're saying we're making MSCs from this iPSC pathway. So we sort of had to prove to the regulators, and initially prove to ourselves, that we are indeed doing that, and that the MSCs we make have at least similar properties to the MSCs that you could get from bone marrow. So that was sort of the initial starting point, and that involved obviously various in vitro testing, looking at cell surface markers, looking at the cells in T cell suppression assays, and so on. And all of that really showed us what we wanted to see, which is that the cells we're making do meet the criteria that have been accepted as defining what an MSC is, but also that they have an ability to suppress T cell proliferation in vitro, for example. And then, of course, beyond that, we had to do a preclinical proof of concept study. So we did studies in mouse GvHD, and again showed very, very encouraging findings there. And the other part of it, though, in terms of the development program, and I suppose a really key issue at the start was, nobody had used iPSC-derived cells in a human when we started out, right? So that wasn't sort of an accepted thing to do yet. Obviously, there was a lot of people pursuing that now. So it wasn't like we were the only people in the world who had thought of it, but certainly nobody had done it. And that meant it was a sort of an interesting regulatory situation because, first of all, understandably, you tend to get some sort of nervousness and conservatism among regulators when something completely new is brought out, but also there was no real kind of precedent or even specific guidelines as to what was going to be necessary. So, we went out and just spoke to the regulators in key jurisdictions really before we had done anything, before we'd even done our preclinical studies, because we just needed to go and understand sort of what they were going to need from us to get into the clinic. And that was very useful. And we did find that we met with obviously the US FDA, but also the EMA in Europe and some of the national agencies there as well, like in UK, Belgium, Germany. And that was a very useful process. And of course, the TGA in Australia, I shouldn't forget them. But that really helped us understand how they saw it and what they would really need from us in order to give us clearance to do an initial clinical trial. And part of it actually was a sort of a unique issue with regard to pluripotent stem cells, which is pluripotent stem cells, as I explained, they're very, very useful for producing other cell types, but you wouldn't really want to administer pluripotent stem cells directly to a human because you wouldn't really be able to predict what they would do. Obviously, they have the capability of differentiating into anything. And, one thing that they can do is, in the right conditions, form teratomas. So clearly, that was not something anybody would want to happen. So what really became key early on was making sure that we had a product that didn't contain undifferentiated pluripotent stem cells. In other words, we needed to be able to say, by the time we give this to a patient, we are administering MSCs, not iPSCs, and not some mixture of iPSCs and MSCs. But the way we did that, and this is really where credit again goes to Igor and his team. Our process, this issue had been sort of really thought about early on, and the process we use is actually designed to ensure that we don't have any iPSCs in the final product. So it's done in a few ways. At one point, there's a mesenchymal colony selection step, filtration step. But then beyond that, we use culture conditions that have been shown, basically that iPSCs do not survive in. So if you have residual undifferentiated iPSCs, they don't survive in the culture conditions that we use towards the end of the process. So that's a very elegant way of ensuring we don't have residual iPSCs. But of course, beyond that, the regulators also wanted us to not just give them this sort of theoretical explanation as to why we don't have residual iPSCs. They wanted us to demonstrate it. So we had to develop our own assay. And again, that assay didn't really exist because nobody had done this before. So that was a as something we had to do, and we developed our own very sensitive assay, which subsequently we've actually patented as well, which is useful. And we also did some experimental work where we did things like spiking undifferentiated iPSCs back into the process at the end and so on, and showing that we couldn't. And so that was sort of a bit of a unique challenge in that it was very specific to the manufacturing process, if you know what I mean. But that was probably the biggest sort of issue that we had to overcome before we could move into a clinical trial. And needless to say, we were able to do that.
It's fascinating because again you're talking about again, this comes back to your pharmaceutical science skills, right? I mean, a lot of this stuff didn't exist, and you got to think, okay, all right, this isn't your traditional small molecule. How do we do it alongside clinical development? So it's very impressive, from that perspective. So I take my hat off to you on that. Going to another really interesting program, because I spent a little time here a couple careers ago, osteoarthritis. You've done some actually late stage clinical work here. Here again, looking at the when we talk about our secretome, obviously we're going to be looking at more of the anti-inflammatory sort of regenerative potential of these cells. But obviously the holy grail in this space is not just signs and symptoms, but cartilage is really hard to regenerate, and great if it comes back and you can get that going. And I know you looked at a couple different endpoints there. Talk a little bit about the Sculptor program and a little bit of where we are in 2025 with how that's progressing.
Yeah. So you're right. We are in a phase 3 trial right now in Australia, which is at an advanced stage in people with osteoarthritis of the knee. So we actually completed enrollment in this trial back in November 2023. It's a 320 patient, fully randomized, double-blind trial. And we have a 2-year follow-up. So the final follow-up visit will be in November this year. So we should have results probably less than a year from now. Which is very, very exciting. But to take a step back in terms of how we got there and what led to it. So this is actually a partnership with the University of Sydney and in particular Professor David Hunter, who is an internationally renowned researcher in the field of osteoarthritis. And he had done some work, as had many others, using donor-derived MSCs to treat osteoarthritis of the knee. So he had done some initial clinical trials, and together with his colleagues there, lots of preclinical research as well. And like many others, had sort of, I guess, seen that there is the potential here for MSCs to have not just a symptomatic relief effect in osteoarthritis, but potentially a disease-modifying effect. So in other words, at least slowing down or stopping the degeneration, potentially even leading to regeneration. That remains to be seen. One thing I think it's important to say is that simply being able to sort of slow down or stop the degeneration would in itself be a huge step forward, because in most cases, people can be identified before things are too bad. So if you could sort of just stop them as they are, that would be usually good enough in many cases. But basically how our program came around then was David wanted to do a proper phase 3 trial like we are doing right now. But when he started to think about this, he kind of realized, well, the way that they were getting MSCs, which was through adipose tissue donations and so on, it was going to be pretty challenging to actually make enough product just even to do the trial, never mind to actually then potentially commercialize a product based on that sort of process. So he became aware of what we were doing and approached us and said he'd be really interested in working together if we could, get some funding to do this trial, and basically asked if Cynata would be willing to supply the product for the trial, which we agreed to do. So this is going back quite a few years now. I think it was way back in sort of 2018 when we started that plan to work together. But the trial itself then commenced sort of the end of 2020. I think the view, and certainly listening to David explaining this, the issue is that there's been lots of work done with MSCs in osteoarthritis, but nobody has really done what would be considered a sort of a gold standard phase 3 trial like what we're doing. So it is obviously really important to do that, because until that's done, there's certainly lots of people reporting benefit, and there's been lots of small studies which have shown benefit, and there's been studies with relatively short duration that have shown very clear effects in terms of symptomatic relief and so on. But does that translate into long-term disease-modifying effect? That's an open question. So the view that David really held strongly was that we really need to do a proper trial like we would do with, again, a typical small molecule drug. What would you do if you had a drug that you thought could help osteoarthritis? Would you go and do proper phase 3 trials like this? So that's what we're doing. And as I said, we're really at an advanced stage now, and very much looking forward to those results. If positive, then, it really is something that would be transformative clearly for us as a company, but also actually for the treatment of osteoarthritis, because we are still in a situation today where there are no actually approved treatments, disease-modifying treatments for osteoarthritis. So at the moment, people take, they take painkillers, anti-inflammatory medication, they get hyaluronic acid or sometimes steroid injections, et cetera, et cetera. And eventually, many of them end up needing a knee replacement. So that's the current reality. And if we could change that, then it would be absolutely transformative.
Absolutely. Absolutely. Yeah. That is clearly, obviously looking at the space, it is one of the holy grails, let's just say, of the pharmaceutical industry overall. So I appreciate your the fact that you're that advanced there, and it's going to be really interesting how that program progresses. One other one I just have to ask you about, because I know it was just announced today, I think. So I hope I'm getting a bit of a scoop here, and whatever you can talk about, that'd be great. You have some preclinical work going on in ischemic heart disease. And clearly, this is the biggest of them all, the major killer of us all in terms of the non-communicable diseases. And yesteryear, we used to just think about sort of, okay, these plaques are just, lipids or whatever. Now we know it's a lot more. We got these macrophages that come in and turn into these foam cells, and the epithelial layer changes. And it's a degenerative process in its own right, that, as you point out, it is immunoregulation, it is inflammation, it is remodeling of tissues. I'd love to hear about what, I know this is earlier stage and it's preclinical work, but I'd love to hear more about this, because I think it clearly, unless we get rid of this one, it's what I call one of the major lifespan killers of us all, along with cancer. And we're not in a great state even in 2025 with it. And I'd love to hear a little bit of where you're going with this, because it's very, very important nonetheless.
Yeah, it absolutely is. And, for the reasons you just outlined, there is certainly reason to believe that MSCs could have a place here in helping to treat ischemic heart disease. And, again, we didn't come up with the idea of using MSCs for ischemic heart disease. There's been, other people have done various trials and so on. The results in clinical trials in particular so far haven't been as good as what people would have hoped, right? And so as always, when that happens, there's questions asked as to why, what is it? And look, again, going back to one thing I mentioned earlier is, the way MSCs work in vivo is really through their secretome, right? So it's the releasing cytokines and so on. But one thing that we also know about MSCs is that they don't persist for very long after they're administered to a human patient. So, and that's an important difference with a lot of other cell therapies. Like, so some other stem cell based therapies, the intention is that the cells that you administer are expected to engraft and basically remain with that patient forever for more, right? MSCs, that does not happen. They get cleared quite quickly, like within a couple of days, typically, if you administer them by intravenous infusion, for example, right? Now, when it comes to a chronic condition like ischemic heart disease, the question then is the persistence of the MSCs long enough to actually have a meaningful effect? And if not, how do you change that? And people have tried all sorts of things, including delivery directly into the heart and so on. But I'm not sure if any of those mechanisms have been particularly successful either. But so the approach that we've taken in this particular study, and I should mention, we ourselves have done previous preclinical work where again we've shown really promising effects of our cells in preclinical models of ischemic heart disease. So we do know that there's potential there. But then this project, it's a collaborative project led by Professor Max Lim at St. Vincent's Institute here in Melbourne. But it's with a really a huge team, not just in Australia, but various institutions overseas as well, including in Singapore and indeed in the US. And the difference in the approach here is that we're actually encapsulating the cells in a device, and it's a device made by a company in the US in Arizona called Procyon Technologies. And what this device does is it protects the cells, so it can keep the cells alive for a prolonged period of time, but it allows the secretome to be released, right? So it's sort of like a sustained release MSC product, which again takes me back to my pharmaceuticals. Yeah. But it's a very clever approach. And what we found, so what was announced yesterday was the results of this initial preclinical study when we've put a manuscript into a journal for peer review. So it's still at an early stage, as you said. But what we found in the study was that a few interesting things. So first of all, when we implant this device where the cells are encapsulated in the device, it was implanted into rats for 12 weeks, and then it can be retrieved. The device can be retrieved, and they found that the cells were still alive and viable after that period of time, which absolutely would not have happened had they just been, infused intravenously. You wouldn't have been able to detect those cells after a day or two if you'd done that. So that was one important point, which really sort of validates the premise that the device can allow this kind of prolonged release of the MSC secretome in vivo. And then beyond that, we found that the encapsulated cells had some very positive effects on the actual disease model. So it was a ischemia reperfusion model of myocardial infarct, and so very widely used model. And the cells did have some really promising effects, as you might expect, on those on the various aspects of that disease model. So it really what it potentially does is offer us a different way to deliver these cells for a condition like ischemic heart disease, which is a chronic condition where it's particularly challenging to get anything to sort of hang around in the heart, obviously, because the heart is pumping. So this is a way perhaps to constantly sort of have the secretome kind of constantly circulating and having effects that way. And so we're definitely interested in seeing if there's a way to bring that into the clinic in due course as well. We haven't to date done any clinical trials in cardiovascular disease, but that certainly is an interesting opportunity for us. And to be honest, it's one of many other opportunities. I mean, as I mentioned really early on, like the MSCs have been investigated for a vast range of conditions, and even we ourselves have undertaken preclinical research in something like 10 to 12 different clinical conditions, and we've published data in many of those, and they've all really been very encouraging. So the limitation for us at the moment really isn't finding new opportunities to use these cells for. It's just like often what the limitation is for small companies, which is you've got to focus your relatively limited resources, both in terms of people's time and energy, and of course money. So, but what we see here is that we've got this year, within the next year, we've got expected readout in a graft-versus-host disease phase 2 trial and our osteoarthritis phase 3. And if they are successful, then clearly that's going to put us in a very different position where we could then be bringing these other opportunities forward as well.
Absolutely. And I appreciate that. I mean, I obviously having hung around the industry, and you wish you could hit every disease possible, and you had $1 trillion in the bank account.
Yeah.
But yeah, I mean, but nonetheless, some of these indications most likely will get you there. And as I said, just for the audience, the company also has ARDS work, diabetic wounds, renal transplantation. Just, while we have you today, I just wanted to ask you about one other thing, and I sort of we're going to looping back to the beginning again, but this one is just quite interesting to me personally, and I just love to get your sort of over the horizon view on something. Just a couple of weeks ago, in this is February 2025, you published a paper. It's entitled "Proteomic Profiling of iPSC and Tissue-Derived MSC Secretomes Reveal Global Signature of Inflammatory Licensing." And in this set of experiments, you basically show that, depending on how we treat these cells, if we expose them to certain sort of inflammatory stimuli, they make more immunomodulatory type substances. If we don't do stuff, more extracellular matrix and regenerative dynamics. Talk a little bit about it, because we've gone from cell, you've talked about cells inside a device. What about just secretomes of these MSCs as drugs themselves? Clearly there's some companies out there that are taking this path, but clearly if you're in a position where you can not just produce 10 to the 72 power of these cells, but lots of bioactive material, I mean, there must be a lot of biologics, protein peptide type opportunities as well. Obviously, you can't go in all directions, but love your sort of over the horizon viewpoint on this part of the potential of what you're developing.
Yeah, and that absolutely is a potential additional opportunity. And you're right, there are companies who have gone down this path, taking the view that, well, if cells work, or if MSCs in particular work by releasing their secretome, maybe we don't need the cells in terms of what we administer to humans. We'll just, isolate the secretome and make a product based on that. And that's certainly an interesting idea. It has some logic. I think there's still some open questions about it, to be honest. The cells, one important thing that we do know about the cells is that in vivo they respond to their environment, right? So how do we reproduce exactly what the MSC secretome is in vivo, in an ex vivo setting, and get that right? That's a challenge in itself. The other thing is, of course, MSCs can migrate to sites of inflammation, for example. We know that. So whereas if it's more of a just administering molecules, well, they're just going to go wherever they go. So there's some questions about that, and whether I don't think anybody necessarily has the answer to all of that, but it's certainly an interesting approach. And I think we are actually very well placed to get involved in that as well, to be honest. If that becomes something that really is, if there's really something to it, because, even if your end product is the secretome of the cells, I think it's still fair to say you want the cells to be consistent, because if the cells aren't consistent, their secretomes won't be consistent either. Yeah. So even if we ended up moving to some extent to a situation where actually for certain indications or certain applications, shall we say, that it's going to be useful to just have a secretome-based product, which might be called, whatever, an exosome-based product or a protein-based product, I think our platform is actually ideally suited for the production of that sort of a product as well. So we're very much open-minded about that. And, we certainly have had some discussions with companies who are pursuing that sort of an approach, and we're very open-minded on that as well. So yeah, again, it's another set of possibilities for us, and that's really a good thing for us. We're not actively pursuing that ourselves right now, but who knows? Watch this space, I suppose. Our view is we're sort of keeping an eye on what's happening, and if there's a good opportunity for us to get involved in something like that, then we're certainly open to it.
Very cool. Very cool. Kelly, what else should we be aware of for 2025? Obviously, you've taken us through an amazing clinical portfolio. Obviously, Biotech 101 is happening as we speak. Yeah. But anything I missed while we have you today, either from the company side or conferences, talks, anything else that's happening public facing that we should know about, please?
Yeah, well, we're certainly, so I suppose the key things, again, just in terms of the major inflection points, we're going to have final results from 2 clinical trials next year, which is phase 2 GvHD and phase 3 osteoarthritis. So certainly keep your eyes out for those. We also have this ongoing renal transplant trial where we'll have some results from our initial cohort probably in the next few months. So that will be really just getting that one started. We also quite recently we completed a clinical trial, a phase 1 trial, as you mentioned, in diabetic foot ulcer patients with really positive results. And so what we're doing, I guess, behind the scenes then is we're out there, we are engaging with potential partners. We are absolutely out there at conferences, and I'm going to be at the International Society of Cell and Gene Therapy meeting in New Orleans next month, for example, and speaking in a panel there about iPSC-based therapies with some really, leading players in the field, including some from Japan. My colleague Matthias Kroll, our Chief Business Officer, will be at Bio in Boston in June, and we regularly go to these conferences, and the aim there is really two things. One is obviously to increase kind of awareness of what we're doing, but also in particular to engage with potential partners. And I think that is definitely remains a key part of our business model. We're not intending to turn ourselves into the kind of company that could be marketing an osteoarthritis treatment worldwide, right? So we think that is better left to the big pharma guys who know how to do that very well. And so that's really our aim, at least with those sorts of big blockbuster indications, that we'll be out there trying to find partners. Obviously, at the right time, has to be at a time where we can do a good deal for the company, but certainly everybody should be aware that that's something that's a process that's sort of ongoing.
Well, clearly these holy grail type products in the hands of the right big pharma partner. Yeah, I mean the sky's the limit. So it's I'm excited for you. I wish you the best for 2025, and as you continue with these clinical programs, and I'm very excited to see how a lot of them turn out. Again, for everybody that's going to be listening to this episode of our show across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Kilian Kelly, Chief Executive Officer and Managing Director, Cynata Therapeutics. We'll put the link to the company in the bio of the episode. But Kilian, I want to thank you for taking the time out to walk us through your programs, tell us everything you've been up to. Thank you for everything you do for all these unmet medical needs. And as we like to say on our show, thanks for helping to create a better tomorrow for literally millions of patients out there. Really an awesome story, and I really wish you the best with the company, and I look forward to seeing what happens in the near future.
Great. Thank you very much. It's been an honor to be on. Thank you.
Great having you.